This video made possible by Neoplants. More about them in a moment. This is a pothos plant. It's one of the most common ornamental houseplants, and you can find them at pretty much any plant store. But what if I told you that this plant, and others like it, could be the future of air purification? Even better, what if the soil itself could also be turned into a living filter that cleans the air? And all of this through the power of genetic modification and directed evolution. Today we're going to learn about how all of this is possible, and so much more. But first, a question. When you use a permanent marker, where does the wet go? I realize this sounds silly, but bear with me. As you know, the solvent evaporates and goes into the air, and at that point we've already long forgotten about it. As a great Australian chemist once said while disposing of a bottle of dichloromethane,
"The atmosphere is nature's bin." No, I can't say that.
But markers like these are only one of many sources of volatile compounds in the home and workplace, and often these invisible gases can be quite toxic. Benzene, toluene, formaldehyde, and more are often part of the industrial processes that are used to manufacture almost everything we interact with. And when we get the final product, they're still slowly off-gassing those toxic volatiles. That new furniture smell when you build something from IKEA, for example, is because of those volatiles coming off your new sofa. And that's not all. Treated wood, art supplies, glues, tape, paint, plastics, all slowly outgassing. As a quick aside, this is also why we have to use certain materials when building vacuum systems. Most plastics literally exude so much gas you physically can't pull a vacuum faster than they can add new gas to the chamber. This is why our plasma coating system was made almost entirely of metal, and the only plastic we used was PTFE Teflon. What's more is that you can just buy a bottle of toluene, xylene, or even in some places benzene and just use it as a paint stripper, producing spectacularly high levels of toxic volatiles. And if you work in a technical field, you're probably exposed to all sorts of nasty things being emitted from the products of your trade. Now, of course, when I say toxic, I don't mean that you're going to die just from sitting on an IKEA couch. It's not the 1800s, they're not painted with arsenic. But breathing junk like this in can raise cancer rates, exacerbate asthma, or just make you feel really sick if the levels get too high. If you've ever tried painting in an enclosed space, you'll know how nauseating and terrible you'll feel from the fumes. Normally you fix this by opening a window, since on average, indoor air is about 5 times worse than the fresh breeze outside. But in a world where the air outside can be worse than the air inside, you're stuck in a box with all the fumes. So, how do we fix this? It's pretty difficult to reform all of industrial manufacturing overnight, especially when we still rely on cheap labor in countries with little to no regulation. So we're going to need a method to remove this junk from the air after the products are already in our homes. But ideally, we want something cheap, renewable, scalable, and if it can clean the outside air too, that's an added bonus. Though the solution to this may come from a place you wouldn't expect. The French. Now, the French are a fascinating bunch. They know how to organize, they definitely know how to eat, and they even invented a machine to check if aristocrats are made out of cake. And Paris, especially, is a truly wild and wonderful place. You can barely walk 100 meters without artists and history and Nobel Prize winners falling out of your pockets. Museums holding so much history are everywhere. A boulangerie to get fresh bread and pastries can be found at regular intervals of about 20 meters down every road, usually followed by a kebab place next door. And for a brief period a few weeks ago, one of the inhabitants of this crazy city was me. I came here to see the amazing people at Neoplants, a company that is changing the way that we think about air quality by using the power of biology. And inside their gorgeous lab space are about a dozen brilliant and passionate biologists. Also, there were literally plants in every single nook and cranny they could physically be stuffed into, which means a huge amount of natural and simulated sunlight everywhere in the lab. And as somebody who identifies as a tropical plant that will wilt in darkness, for me, this was fantastic. We're going to take a trip through the various processes and techniques that they use to modify both bacteria and plants and see the fascinating and beautiful things that they've cooked up. Also, something that was said many times throughout my visit was that testing and proving that your genetic modification works as intended is often much more difficult and time-consuming than making the modification itself. and as a fellow genetic engineer, I wholeheartedly agree. So the team has put a huge amount of effort into building a super custom testing setup that doesn't really exist anywhere else in the world, but we'll get to that in a bit. Now, you may have been as confused as I initially was when you heard that they're doing genetic engineering work in Paris. Europe is pretty famously against all things GMO, often to their own detriment. But while there are definitely a bunch of extra safety precautions they have to take to do the research here, it's only the sale of GMOs that is, for all intents and purposes, prohibited in Europe. But if they can't sell genetically modified plants in Europe and will limit those to countries like the US with less intense regulation, they still wanted to make something that could be sold anywhere that accomplishes a lot of the same goal. And for that, they cooked up the first product that they released on the market, Power Drops. Now, the idea behind these are pretty simple. First, they screened soil microbes to find ones which are non-toxic, non-pathogenic, fast-growing, and easy to culture. Then, those that they found were further screened for their ability to break down toxic volatiles. Finally, they were mutated and put through a process called directed evolution to improve their ability to break down those volatiles. The result is a powder that can be mixed with water and then added to the soil of your houseplants to massively improve their ability to uptake and start cleaning the air. To make it work even better, they developed the Neoplants. It's their all-in-one air purifier where you have a spot for the plant and power drops, and then a special shell to increase airflow. And while we're going to talk about how they test it to make sure it's working in a minute, first I want to talk about how they actually made the bacteria. That last step of directed evolution is actually an incredibly powerful technique. I've often said that biology was humanity's first and will be humanity's last technology. And directed evolution was the first tool that we unlocked all the way at the start, 10 or 11,000 years ago. Directed evolution is very similar to normal evolution, only much faster, and we're in control. In nature, evolution only has one rule. To paraphrase Darwin, you can't fuck if you're dead. As in, if you die before you breed, your genes won't transfer. But otherwise, nothing is forbidden. When you combine this one rule with the inherent instability of DNA and the fact that it naturally mutates between generations, you end up with a self-correcting cycle. The environment slowly changes, and in turn, organisms living in that environment slowly change. Each generation producing a number of descendants, some of them dying and some of them surviving to breed, and mutating a little bit each time. Over millions of years, organisms can take on wildly different forms as they, like clay shaped by time and the environment, morph to better fit whatever niche they can exploit to survive. Directed evolution is actually nearly identical, only we control the environmental conditions, and we ideally choose an organism that replicate themselves every 20 minutes. So many generations will pass in the course of a few days. Though it does also work with other species if you're willing to put in the time. And by choosing the right conditions, we control how that species morphs. Take sheep, for example. It may have taken several thousand years, but we took the Asian mouflon and morphed it into a fluffy thing that grows so much hair it will literally die if we don't shear it. Or some varieties have a tail so large and fatty, we have to provide it with a cart so it can carry around its own ass. Ironically, even though the process is slow, it does directly change the DNA of the organisms we evolve, and in some cases really dramatically. As I mentioned in a recent video about genetic engineering, kale, kohlrabi, cauliflower, broccoli, Brussels sprouts, turnips, cabbage, gailan, collard greens, and mustard are the same plant. Different groups of humans just selected for different attributes they liked. So, technically, every single crop and farmed animal sold in Europe is genetically modified. But because it's done randomly and slowly, for some reason they allow it. Whereas fast, precise mutations are disallowed, which is frankly a twisted knot of dream logic that to me makes no sense, but that's the rule. Though I do understand that often it's not really the dislike of GMOs per se, but rather the desire to protect otherwise wonderful heirloom strains that would be outcompeted. Still though, the rule's a bit silly. Anyway, the point is that given enough time, you can make enormous changes to an organism. But by working with bacteria that have a doubling time of 20 minutes, you can do a lot with a few days or weeks, instead of needing hundreds or thousands of years. When they grow that fast, 500 generations can happen in just the span of a week. The mechanics of the process are actually incredibly simple, though there are a few ways to go about it. The most basic method uses a bioreactor system like this one. This particular machine happens to have been built by a very good friend of mine, Sebastian, and was nicknamed the Baby Volver. The idea is that you start with a culture of your bacteria and then slowly add in fresh food while withdrawing the spent media. By slowly changing the composition of the media to include more of your target compound, you'll encourage the growing microbes to mutate, and only those that can survive the current concentration will grow and divide. Over a surprisingly short time, you can end up with microbes that are wildly different from the starting culture and much better adapted to your target conditions. But sometimes the microbes need a little push, and the natural mutation rate just isn't enough. For that, you bust out the big guns, and by that I mean we irradiate the hell out of them and scramble their DNA a bit on purpose. This is done in a UV box like this one. You start by putting a freshly inoculated lawn of bacteria into the box with the lid off. The lid would otherwise prevent the UV from penetrating. Then all the bacteria need is about 3 seconds of UV. This is enough to kill the vast majority of the cells, and the ones that are left will have their DNA a little bit jumbled, but not too much. Bacteria are remarkably resilient little critters and can handle quite a bit of DNA damage. Then the bacteria are exposed to the volatile of choice by making a little reservoir out of agar on the inside of the lid of the petri dish, and then putting a little bit of toluene or whatever else you're interested in on that agar. This way, the microbes will be constantly exposed to the vapor, and the mutants that grow the fastest and show the most resistance and degradation of the chosen volatile is your winner. You can repeat this process a few times, and again, very quickly, you can direct the evolution of the microbe to be much more useful than its original state. That said, while directed evolution is amazing, it's hardly precise, and you can't directly add new genes like we've explored in a recent video. So let's move on to their more advanced work and finally see some of their incredible plant labs. Plants, as you may know, are super weird organisms. Because they can't get up and change their location, their genetics include a lot of complexity to deal with potential situations that might arise at some point in their life. And even their way of growing is very different than what we're used to in animals or microbes. For all intents and purposes, plants are the expression of hormone distribution over time and space. Everything a plant does is because of some hormone or chemical gradient as it grows and responds to its environment. Because of this, plant cells can reprogram themselves at any time to do something else if you provide them the correct hormones. This means that if you take a chunk of sterilized plant tissue, like a bit of leaf or stem, and then plate it on the right media, you can get it to form what looks like a little booger of plant stem cells. This booger can then be moved between other kinds of media to first induce shoots to form, and then roots, producing a brand new baby plant ready to be moved to soil. And being able to do this is absolutely critical if you want to be able to genetically modify your plant of interest. But of course, as there always is, there's a problem. Every plant, and even just different strains of the same plant, will respond to hormones differently. So getting the witch's brew that goes into the media correct can take a long time. On top of that, plants grow really slowly, so to go from these callus-like boogers to baby plants takes about a year in some cases. And in the case of pothos plants like these, 28 different kinds of media over that time. It was also very cool to see examples of plants being cultured that very few others have attempted, like Monstera, another classic and beautiful houseplant that is famously hard to culture. But once you've got that working, you can actually do the fun part of genetically modifying the plants. So, how do we get new DNA into our plants? Well, we shoot it with a tiny, tiny shotgun. And yes, I am 100% serious. The machine is called a gene gun and is easily my favorite tool in the whole lab. Instead of steel balls or lead shot, the bullets are made out of tiny gold nanoparticles. The idea is that you take the gold nanoparticles and incubate them with DNA in a special solution that makes the DNA stick to the gold. This is then dried onto a burst disc made out of what I think is some sort of Captain-like material. Though I will say, I asked Iman, their head of plant biology and operator of the gene gun, to really overload the samples so that this is easier to see. Normally, you use a lot less gold than this. Once the solution is dried, it's placed on top of a mesh screen in a special carrier plate. Then a second burst disc is prepared, but this one has no gold on it, and it sits higher up in the machine. This second disc acts as a seal between a helium reservoir and the vacuum chamber that is the main body of the gene gun. The idea is that the chamber is vacuumed out and the reservoir is pressurized with a specific amount of helium. When the helium is allowed to push up against the burst disc, it will suddenly fail at a specific pressure. This produces an incredibly fast jet of sterile gas that starts flying towards the lower burst disc that has the gold on it. On impact, the gold is forced through the mesh at incredibly high speed, bursting tiny holes in the Kapton barrier and then flying towards a leaf sample that's placed at the base of the chamber. The gold embeds itself into the leaf tissue, and some of those gold bullets will land inside the nucleus of the plant cell, delivering the DNA. Iman told me that while the main impact site will give you really good transient expression of your gene of interest, you actually tend to find the best clones that have integrated the DNA properly further away from the main impact. And again, because we loaded a ton of extra gold, you can actually see where that main impact site is, but the whole leaf is covered in tiny holes. These bits of leaf can then be subjected to the callus forming process, and with the addition of a chemical to kill off any of the unmodified cells, you've got the beginnings of a modified plant. But for all this to work, you need to have DNA to load into the gene gun in the first place, and to that end, a number of the rooms in the lab are dedicated specifically to molecular biology and building their DNA constructs. They start by designing the DNA on the computer like we showed in a recent video, and if you want to learn how to design DNA yourself, there'll be a link in the cards and below. Sometimes the bits of DNA they use are extracted from specific organisms, and sometimes they're printed from scratch. They then take all of the little pieces that they need and cut and paste them together into the final construct. And since they do this at such large scale, running dozens of experiments on any number of projects, they use tools like this cool Opentron robot to do a lot of the tedious manual labor. I once made a video showing off these amazing robots and got mine to make petri dish art. But instead of painting on a canvas of agar, Neoplants paints on a canvas that is living plants. There are all sorts of changes that they can make to the plants, be it expressing custom-designed proteins, tweaking their metabolism, or either increasing the expression of certain genes, or knocking others out. For the most part, this just ends up looking like the same as the parent plants, but their current plants remove up to 30 times more volatiles from the air than the unmodified ones, and they're constantly making tweaks to improve that even further. Because of the controls on genetically modified plants, all of the adult plants that are growing in soil must be kept in a special double-layered, hermetically sealed vault. And every single leaf that falls or is removed from one of the plants has to be disposed of in a special waste bin that is then carefully treated by a specially approved company. All of this to say, they take their biosafety here very seriously. But hidden in these different rooms are some really wonderful creations. My favorite are these amazing red pothos. It's a little side project they're working on, and there are a bunch of different versions. Some are dark red, others are beautiful light pink, and some only turn pink when the leaves begin to wilt. But of course, those are just the obvious ones. The rest, while they still look beautiful, just look like really well cared for pothos plants. But inside, they hide all kinds of genetic tweaks and modifications that make them much better at cleaning the air. Speaking of clean air, as you can imagine, they take air quality really seriously, and with a room like the plant vault, they are required to have incredible control over air quality to make sure nothing comes in or goes out that isn't supposed to. To that end, they have 4 of the largest air filter machines I've ever seen in my life. For scale, I'm 5'10". I barely came up to half the height of this thing, and again, there are 4 of these monsters. The great irony is that they're based in a city that smokes approximately a bajillion cigarettes a day, so the air quality outside of the building was terrible. Being in the lab was quite literally a breath of fresh air. Now, these mechanical filters are lovely, but let's now move on to talk about how they prove their plants and power drops really do what they're supposed to. They have essentially 4 levels of testing that start at the small scale of molecules and end in 2 specially made rooms that cost about $1 million to construct and install. Analysis starts, as all good analysis should, with a bucket of liquid nitrogen. First, some liquid nitrogen is casually scooped into a mortar and pestle along with the plant material they want to test. In this case, a leaf from one of their modified plants. The flash-frozen tissue crumbles to dust when grinding, and you're left with a perfect powder ready for further processing. Next, the powdered leaf material is treated with a solvent to extract out the chemicals that they're interested in, and the choice of solvent will depend on what they want to study. The compounds in the leaf material dissolve super fast because of how finely divided the powdered tissue was, leaving a vibrant green solution. This is centrifuged to remove any debris, and the liquid can be transferred to a vial and loaded into one of their amazing mass spectrometers. As expensive robots go, a GCMS system is certainly one of the most sci-fi. It goes through this little dance of preparing the needle and then the sample before injecting a tiny amount of liquid into the machine for analysis, and it will spit out a huge amount of critical information about how the plant was performing. Of course, there are other biochemical tests that they run on their plants, be it protein gels or PCR or other assays, but the GCMS room is probably my favorite, if only because it's such a shiny toy and one that I've frankly never gotten to play with. That, and I don't get any PTSD flashbacks the way I do from protein gels or PCR. I mean, it is called pipette, cry, repeat for a reason. Anyway, assuming the plants pass all of these basic tests, it's on to see if they can actually clean the air. They do this at 3 scales: jar scale, 30 liter, and room scale. And as they go up in scale, they also go up in precision. The small scale tests come in 2 parts. The Sentinels, which are these cute little jars that can detect things like benzene and toluene, and then separately they have a whole specialized system specifically to check for formaldehyde, which turns out to be its own hassle. The way the Sentinels work is that you put a plant in the jar, and then there's a special sensor in the lid of the jar that measures airborne volatiles. When all the jars are basically ready to go, they prepare a flask with a little bit of their choice volatile. In this case, they use toluene. The flask is heated to convert the liquid to gas, and then a set amount of that gas is carefully captured and injected into one of the jars. The jar is then quickly sealed and transferred to one of the base stations in the cabinet of base stations. This is repeated for all the jars in different test conditions. So some will have control plants, some will have modified ones. Then, as the test runs, they measure how quickly and effectively they are at removing the solvent from the air. The formaldehyde system is similar, but with some key differences. Firstly, formaldehyde tends to dissolve very quickly in water, so the plants have to be carefully wrapped with cling film to isolate the soil and the actual plant bits. They had to try many different materials to find ones that worked without off-gassing a ton of formaldehyde, but still lets the plant breathe, and this is what they found that worked best. Then the plant is placed in one of the special jars, just like before, but these jars are connected to a special air line that allows you to mix in a specific amount of formaldehyde. This flows through the jar and out a vent into a special gas transfer bag. When the bag is full of gas, it can be detached and then connected to the inlet of their special formaldehyde measurement machine. Assuming the plants pass all of these tests, they move up a level to the much larger jars. The same basic tests can be run, but now the sample of gases can also be taken over to the GCMS room for careful analysis. And the bigger the volume, the slower the test and the more realistic it is. This 30-liter scale is currently their second largest scale, and after this they move on to the special rooms. But jumping from 30 liters to an entire room is kind of a bit much, so they're in the process of building a giant steel box that is somewhere in between the two to allow for still large-scale testing, but a little bit faster. Though since that wasn't finished when I visited, let's talk about the special VOC rooms. Floor to ceiling, the entire room is made out of entirely stainless steel and a special Teflon sealant. It's all computer controlled, and there are 2 identical rooms so that they can have a control to compare against. From the rooms, long lines of Teflon tubes stretch to the dedicated analysis machine. Apparently, there are only 3 or 4 of these in the whole country, and it's a full mass spectrometer, so it gives complete analysis of what's in the air, not just a general VOC level. They told me that if I farted in one of the rooms, they could probably tell me what I'd eaten for the last week, but joke's on them, it was entirely kebab. But to me, all I could think of standing in that incredibly expensive space was the room temperature room from Community.
Feel that?
Actually, I don't feel...
Precisely. Have you heard the expression room temperature?
Of course.
This is the room.
Even though the rooms are hermetically sealed, they still need to tape the door shut just to make absolutely sure nothing gets through, and importantly, that no humans walk inside while they're running one of their month-long tests. They test all kinds of things in here, from fresh furniture, pens, inks, incense, and of course, all of their different air filtration methods, be it plant or enhanced soil. And only after passing this test and definitively demonstrating that the modified plants and microbes clean the air in a whole room are they considered for sale. Thus far, they've been able to achieve a 30% reduction in airborne volatiles with their plants and power drops in their 160 square foot test space, which is amazing for a few otherwise innocuous plants that you'd normally just think of as background decoration. I really was incredibly impressed with their setup and the clear attention to detail to make sure that their products really do what they say on the tin. I'll admit that when I first heard about the company and their products, I was a little bit skeptical. I mean, probiotics for soil sounded like something that Joe Rogan would come up with after railing a line of protein powder. But it actually works, and not only is the science good, it's great. And after coming here, meeting the team, and seeing the hard work and absolute genius on display, that skepticism was comfortably put to rest. And it's so refreshing to have a plant genetic engineering company that really does care about the products and the environment, and aren't just a bunch of suits doing their best to make Temu brand Monsanto. If you want to follow along with Neoplants' journey, or just try the power drops for yourself, head to neoplants.com. And if you use the coupon code ThoughtEmporium, you'll get 10% off your order. As always, I also need to say a special thank you to our amazing patrons and channel members that help make these videos possible. And if you'd like to help keep the flow of science videos coming, or get your hands on some sweet mad science swag, there'll be links in the description. That's all for now, and we'll see you next time.